All right, everyone, hello. Everyone on Zoom, hi. I hope you can hear me. We have some folks in the room and a number of folks on Zoom. So thanks for joining. And let's get started so we can go outside and enjoy the rest of the afternoon, hopefully. Appreciate everyone joining in to hear about what we're doing in MMP. This is one of our newer projects, and I'm excited to share some of our results and what we're doing down in MMP using DNA. Before I start with the good stuff, I'd like to acknowledge some people. First, this is all funded by the Commission, so big thanks to the Commission for continual support since 2019, annual support for our DNA monitoring. As you'll see, it's a collaborative effort, and SRBC fits in really well as a agency to take care of a lot of this monitoring. A lot of it crosses state borders, and it's an easy fit for us to do since we can kind of come and go like state agencies can't quite do as well. Our old friend and colleague Matt Shank, the DEP defector, this was his original idea. So shout out to him. Since then, myself and Aaron have been co-leading this effort in kind of different ways a little bit. If you want to know more about fish passage and how this impacts hydrodynamic licensing and fish passage, talk to Aaron. He's expert on that. We're pretty lucky. We have a world-class genetics lab right here in our basin. Fish and Wildlife Service has a lab in Lamar, Pennsylvania, that does, I think, 95% of the genetics work in kind of the East Coast region. So they're highly qualified, and we're lucky to have them so close, and they've been our partner in all this work. Other field help, other folks have helped in the field over the last 3 or 4 years, Andy King, Josh Inners, Blake, Matt, and Joanna. So big shout out to everyone who helped out. Let me first start out with some easy questions here, some background. What is eDNA? I'm sure you've heard of DNA, but what is eDNA? The E just means environment or environmental. So it's DNA found in the environment. Typically that means air, soil, or water. DNA, if you think back to your genetics class, is a subcellular fingerprint and of a base pair of nucleic acids. These things might ring a bell. But basically, it's a way to identify you as you, and in our case, a fish species as that fish species, using just the DNA fingerprint. I'm going to talk briefly about 2 ways that eDNA is used, kind of in this field, environmental DNA. Species targeting, which is what we're doing. We're looking for a presence-absence result for a specific species. So in our case, say northern snakehead. Is northern snakehead DNA in this water sample, yes or no? That's one kind of eDNA application. The second kind of application is more broad, not as reliable yet. It's called meta-barcoding, and basically you would take a water sample and the lab would throw it against everything they have in their genetics bank and say, these 20 fish are probably in here, kind of thing. We have done some work with EPA and the National Survey to try to help kind of forward this method a little bit. It's not very reliable. For instance, we did a sample in Penns Creek in 2019, and we did a fish assemblage sampling by electrofishing typical methods. We got about 19 fish species. They ran through DNA barcoding stuff, and it said there was 8 species there. So it's not as reliable as just saying, is this one species here, yes or no? But it's getting there. It's getting better, but it's not quite there yet as a method to replace like an actual fish sample. Or and they are using it for macroinvertebrates too, but that's obviously a lot more species and a lot smaller sort of biome. So eDNA is super useful and super important because we can cover a large area, geographic area, quickly, and it's great for a screening tool. So for instance, if you've ever been with us or seen us do a electrofishing survey, it might take 4 or 5 hours for a team of 4 people to do a medium-sized stream. So you can maybe do 2 in a day if you're lucky, if they're close by. We can sample 25 sites to 30 sites in a day easily to get yes or no on these key species. And if you're looking for a specific species that's rare, you might not even find it. If you fish for 4 hours in a stream, if it's only a couple in the whole stream, you might even find it. But this is a better way to quickly see kind of the geographic extent of a target species. The eDNA comes from skin cells, waste products, mucus, scales, like that, things like that, and that might slough off the fish in any capacity. It does not stay in the water column very long, so the sample you take pretty accurately reflects what's there recently or currently. So it's not like some could have been there 2 months ago or 3 years ago and it'd still be there. It's pretty transient. So now the other piece is invasive fish species, and why do we care about invasive fish species? Well, there's no native predators. There's no knowledge on how they'll impact ecosystems that are existing, food chain disruptions, potential to overtake and decimate native fish populations. If you've ever seen any Asian carp videos in the Midwest when they put a shuck in the water and there's millions of Asian carp come up, we don't want that in Susquehanna. We're trying to avoid those kind of dominant species, either biomass or numbers, which is one of the things that this sort of fish can do to an ecosystem. And also they can bring new diseases to a watershed that weren't here naturally and natively. So there's certainly something to be concerned about. And if you heard Aaron's talk at Wildwood on Tuesday evening, he mentioned the difference between non-native and invasive as more of a behavioral thing. So non-native, like you might catch a goldfish, that's non-native, but it's not invading the system. There's not taking over anything. But something like northern snakehead is more an invasive behavior. It's non-native as well, invasive behavior, as it has the potential to take over an entire ecosystem if it gets out of control. So what's the process for this eDNA analysis? It's pretty straightforward on our end. We certainly have the macro level things and the easier parts. We collect a 2-liter water sample out of a stream or a river. You can see Aaron doing that there. It's more important than our typical water sampling that we be very sterile. You don't want to transfer DNA from one place to another. You can't see DNA, so we have to be pretty careful. Lots of gloves are used, lots of Clorox wipes to keep everything clean and not to contaminate our controls and not to contaminate one sample with another sample. So collect a 2-liter sample of water, put it on ice. Within 24 hours, we sample that whole 2-liter. We filter the whole 2-liter sample through a very tiny filter. It's about the size of a $half, but the pore size is 1.5 microns. So for 1 micron is a 1/1000th of a millimeter. So it's a very, very small pore size. The DNA gets caught on the filter. So in theory, all the DNA in that water will be caught on that filter. We put it in a little tiny vial and then label it and freeze it. And our part's really done at that point, which is pretty simple and straightforward if we can keep everything clean. This next part, the DNA genetics part, is a little more complicated, and I don't understand every part of it. I'm not a geneticist. I'll try to explain a little bit how they do things at the lab in kind of the most general sense to give you an idea of how it works. So they have a filter that's frozen in a little tiny vial. They thaw it, use some chemicals to extract the DNA from the filter. And they take a tiny little, like, micron or two of water, microliter of water. So they take 8 little samples of water from each filter extraction. They put them in these little tiny wells. And then they have what's called a DNA marker that is made so that it matches the DNA you're looking for, the target. So there's a different marker for snakehead, different marker for goby, different marker for blue catfish. And so if you're looking for a snakehead, you put the snakehead marker in each of these little 8 replicates from a single site. And if the DNA of the target species is in there, the marker makes that DNA replicate. So it starts to amplify the DNA, and they run it through a machine, and if it's there, it will show up as amplified, and you'll see the copies of the DNA are present. I know it's confusing, but trust me, it's confusing to me too. because it's all obviously, you can't see it. It's microliters of water, and it runs through a machine. But the DNA replicates until it's able to be seen on a screen. Basically, it has enough DNA to be shown on their output screen. And I'll show an example at the end of what that might look like as far as how much DNA is in the sample and what that might look like as far as magnitude of DNA available. So they give us presence, absence, so yes or no. How many of the 8 replicates was positive for the DNA of the fish? So sometimes all 8 were positive. Sometimes it's only in a couple of the 8 replicates. But any of the 8 replicates counts as a positive hit for that site. And a little more in the weeds, but they also report to us what's called cycle threshold, which is basically how quickly the DNA is visible in their systems, or how fast it replicates enough to be seen. And typically, if it doesn't show up to after 40 cycles, they say it's not there. So typically, you see between 32 and 38 kind of cycles to see amplification. If it's less than 30, there's tons of DNA in that sample, and so you can use that cycle threshold as kind of a surrogate for how much DNA is there. They also report DNA copies per liter, so you can get a handle on kind of density of the fish in that sample. I'm going to talk about 2 different case studies. They're separate but different, 2 different geographic areas, a little overlap in the fish species, but I'll go through them separately because they're 2 separate efforts. I don't want to get confused here. So the Upper Susquehanna effort, I'll talk about first. We looked for round goby and northern snakehead. And so here is the study area. The map you see is the Upper Susquehanna. The yellow outline is kind of the general study area. In 2019, I was made aware of kind of a need through some of our New York partnerships for monitoring of invasive fish species in this region, especially the part that overlaps with the greater Catskill regions, which is Otsego County, Delaware County, and Schoharie County in the Susquehanna. So this area right over here especially. So the commission generously funded this study, and we sampled 60 locations in October of 2020, looking, like I said, for the presence of brown goby and northern snakehead. So why these fish? So the brown goby first, it's a little guy, it's a benthic fish, 4 to 6 inches long approximately. Looks like a sculpin if you're a fisherman. Native to Eurasia. It's considered invasive because it spreads very quickly. It was probably introduced through ballast water in the Great Lakes, and then through bait buckets, other places. We did have one record. A dead goby was found in Binghamton, which is kind of where that little arrow just popped up at, around that area. In the Susquehanna, that's the only record that we know of so far. And it was dead, but it was found there. It was a verified record. However, the brown gobies have recently, in the last 5 years, spread quickly in the Mohawk River Basin, which is just the basin north of Susquehanna. It's shown in that black circle there. And they went from a lake through some canals to the Mohawk River, and they're now in the Hudson River in New York State, all in the last 5 or so years. And the USGS up there has been doing eDNA monitoring with them, kind of following their invasion the whole way down to the Hudson River, which I think just was confirmed less than a year ago. And so it's not hard to make a assumption why right here in Susquehanna is kind of a high-risk invasion spot based on proximity to the Mohawk. The northern snakehead, probably have heard a lot more about. On the news in the last couple of years, they've been called the Frankenfish, and they're gonna, change everything and kill all the fish. And I think they'll ask me if they're harmful to humans, and I'm not gonna come out and attack you. Certainly, they are certainly a potential dangerous introduction to the ecosystem of our native fish. They have voracious predators. They multiply, reproduce numerous times per year. They can travel over land for short distances. They're very hard to kill, but they're also a good eating fish and a fun fish to fish for. So they have been spread, I think, intentionally and accidentally. And there are some records of snakeheads in the upper Delaware, which is obviously right next to our basin to the east. And so they're concerned about snakeheads getting to upper Susquehanna as well. These pictures here are some we have caught ourselves. This one, the top small one there, is from the basin in Deer Creek. Little one, and this one's actually in the Susquehanna River in the Delaware Basin. We sampled it for NERSA in 2019. They can get really big. So the good news and the bad news, I guess, if you want good or fun results. But the good news is there was no hits of either species at all at any of the 60 sites. It was all clear, no goby, no snakehead throughout any of these 60 sites. Those green dots, we sampled all those locations over a period of about 2.5 days, and all clear. And the lab was even concerned maybe the DNA wasn't valid or viable. So they did run the DNA against smallmouth bass, and there was 95% of the sites had hits. So we know it was good DNA, good samples, but just no round gobies or snakehead, which made us more confident in our results since they did check the DNA for viability and that kind of thing. We didn't just give them like sink water or anything. We gave them actual stream water. So there's no round gobies, no snakeheads. Great news. Aaron's part of the round goby task force, and a lot of the DEC coordination with other fisheries professionals. So he's keeping tabs on round goby as far as that. That's probably the more likely invader at this point in the near term. But we definitely would like to keep monitoring every 3 or 4 years to kind of make sure we're keeping tabs on what's happening up there in New York State with these two species.
Hey, Luanne, can I ask you a question?
Sure.
Is there anybody else using eDNA to monitor up there, or is it just Susquehanna River Basin Commission?
I think right now it's just us. There was, DEC was kind of leery about us doing it because someone had a false positive that they couldn't verify or validate, and so there was some skepticism about using eDNA. That was probably 5-6 years ago. I think it was with snakeheads, and they never found any substantiated extra samples. So I think we're the only ones doing it in that area right now.
Thank you.
All right. That's the case study number 1, Upper Susquehanna, way far away. The next case study is a lot closer to home, and you may have heard a little bit more about this as it's maybe been in the news, or at least been kind of spoken about in the office a little bit more, I think. Lower Susquehanna, as everyone knows, is complex, and it also has 4 major dams in Lower Susquehanna. As you see in this map here, there's Harrisburg, and we have 4 here. Conowingo Dam, which is in Maryland, which is in the picture below. It's 90 or so feet high, impassable, obviously. Holtwood, Safe Harbor, York Haven is the top dam just south of Harrisburg. So those 4 dams, they prevent natural fish passage of things like shad and American eels who are trying to migrate, and they also can keep out invasive species, which is good. But as we're trying to pass native fish we want to pass, we have to be careful not to pass other fish species that we don't want to be in the river. And so Matt Cheng started this project in 2019. We sampled 19, 20, and 21, and we have plans to continue in 22 here this spring. Again, looking at northern snakehead, we know northern snakehead already below Conowingo Dam. They're in the bay. They're in the other tributaries that come into the bay. They come up the bay to Susquehanna. They're caught below this dam pretty frequently. Second species is blue catfish, which are caught below the dam as well by anglers. They have not, as of yet, been caught in the fish lift that they use to pass fish over Comingo Dam. Because some of our work and Aaron's involvement with the fish passage team, in 2021, they started sorting every fish that comes to the fish lift on the west side of the dam. So the west fish lift is over here, the far side, and so fish come up to that, and they come into this mechanism, and usually they would just take all the fish and toss them over the dam. But now we make them sort every fish. I think 2021 they sorted out 950 snakeheads, over 1,200 maybe flathead catfish, but no blue catfish were found in that system. What else about the general study? Oh, in 2020 there were unintentionally, before they started sorting, about 24 snakeheads that people were watching them pass the window, and they're like, oh crap, there's 24 snakeheads, and they were already over the dam. So we knew that there are snakeheads above the dam there in Comingo Pond, but in 2020 we did not catch any DNA in our samples, and you'll see in this past year we did actually find some DNA above the dam there for snakeheads. So that's kind of the general overview of the project and the whys and the whats. Talked about snakeheads already, so what about blue catfish? We have a native catfish called a channel catfish. The blue catfish looks pretty much like a channel catfish when it's small. Channel cats don't get this big. We also have an invasive species called a flathead catfish, which is pretty much ubiquitous across Susquehanna, and it's kind of a lost cause effort to try to keep those under wraps. You can find them from Harrisburg to Wilkes-Barre pretty easily, and in tributaries too. But the blue catfish, used to be river drainage fish. It's in the bay, everywhere in the bay, and it's coming up to Susquehanna. As you'll see, they're pretty much all over the river below the dam. Their DNA is anyway. They'll eat almost anything. I just told Mike earlier, there's a whole wood duck was eaten by a blue catfish. There's been an entire wood duck in the stomach of a blue catfish. They'll eat mammals, they'll eat blue crabs, they'll eat fish, they'll eat just about anything. And they can get very large, as you can see, and they have a potential to take over like a big, huge biomass and disruptive in the native food chain. But they are fun to fish for, apparently, so people are not so excited about getting rid of them or keeping them away. All right. I'm going to try to open up this nice interactive map that Aaron made. Oh, it worked just like that, Mike. How about that? Thanks. So shout out to Aaron for making this map interactive. He used it for some of his presentations. So if you've already seen it, I apologize, but I think it's the best way to show kind of the progression of this project and as well as the results. So just to give you some context, Harrisburg's here. Here's the 4 dams. There's the head of the Chesapeake Bay right here, Conowingo, Holtwood, Safe Harbor, York Haven. So let's start out with just showing the sites that we sampled. One thing I don't think I mentioned was that there are 2 main tributaries that come into Susquehanna below Comingo Dam. Deer Creek on the west side here, and Octoraro Creek on the east side here. And they sampled those pretty heavily to see how far snakeheads and or blue cats had gotten. So this is 2019, this is our first year. We targeted very heavily below Comingo Dam in Susquehanna. You can see we did transect sampling, so the whole way, so 4 samples across the river. It's very wide here. We wanted to know kind of spatially, if they were in certain areas or with certain tribs, but we sampled all, so 4 across all these transects, and then the whole way up Octoraro Creek. Right here's Octoraro Reservoir, sampled above Octoraro Reservoir and below it, and this is Deer Creek. Small Lowhead Dam about right here. We sampled below that Lowhead Dam, and then above it the whole way up to the top here. If you know where Eden Mill Nature Center is, that's way up here at the top of Deer Creek. So that's our first kind of study region for this. In 2020, we added some more sites. We moved upstream a little bit. We lessened our sampling intensity below the dam because we know they're there already, and we wanted to make the most of our resources and sort of stay ahead of the invasion front. So we added more samples in Conwingo Pond, and we added a couple sites even above the next dam up, which is Holtwood, a couple sites here. We're kind of, using the adaptive management plan strategy to kind of adapt as we go along. And then this past year, 2021, we added a lot more sites, more upstream, so the whole way up, even above York Haven, which is the last dam upstream. We had a record of a snakehead being caught in a quarry over in an old quarry, which is now in a park in Ephrata. So we sampled there. And we also, if you've ever driven down 83 past York, you know Lake Redmond's right there. And there's been some records and some pretty valid reports of snakeheads in that system there. And so we sampled kind of the inflows and outflows of those two reservoirs together. So as you can see, we kind of moved upriver. So we have data from about the I-95 bridge crossing down here, right above the head of the bay, the whole way above York Haven. Those are just our sites. Start with some results. So northern snakehead. This is just northern snakeheads and just a positive hit. So this is if any site was any sample was positive any of the 3 years, you'll see it on here. So let me zoom in lower here a little bit. You can see pretty common across some of the transect sites, not all of them, but some of them. Deer Creek mouth is right there. Definitely a positive hit at Deer Creek mouth. Octoraro Creek the whole way up the base of the dam. There was none above the reservoir as of yet. We do know that there was snakeheads caught in Octoraro Reservoir last summer, a number of them, 9 or 10 last I heard. So probably only eventually a matter of time till they get above. But as of last spring, there was no DNA detected above Octoraro Reservoir. You'll see here, first, these two are from 2021. This is like hot off the press data. Here, this is Broad Creek that enters Conwingo Pond on the west. We had a northern snakehead hit at the mouth of Broad Creek, and also in the river just below Broad Creek mouth. So this is the first time we found DNA of snakeheads above Conwingo Dam. We knew they were there in very small numbers, but we did capture their DNA in 2021. And I'll just put on, just for completion's sake, kind of, and there was DNA in this quarry up here. There was snakeheads in that quarry. It's isolated. There's no inflow and outflow, so someone put them near the fish is what probably happened. So these yellow dots here are all the native snakehead sites, which is good. Like I said, they're not above the reservoir in Octoraro as of yet. They're not above that small Lowhead Dam on Deer Creek as of yet. That's all negative on the main part of Deer Creek, upstream of that small dam. There's none above Holtwood or Safe Harbor or York Haven. So that's great. That's good news. They are contained right now in Conwingo Pond and below. But we're going to keep monitoring, and we'll probably do some revisions to our sample design for this coming up year to kind of keep ahead of the invasion and focus on some of these areas a little more intensely. Maybe move more points into the pond and try to see what we can find. All right, let me go quickly to blue catfish. All right. So this is positive blue catfish. So let me zoom in. I mentioned before they're everywhere, so every single transect at every single spot across the river, the blue cat hits the whole way up. So they're very prevalent in this lower part of the river. It's not surprising because they're in the bay everywhere. So they've come the whole way up here. We're unsure as to why we've never found them in the fish lift. Maybe they're not interested in migrating upstream that much. Maybe the noises deter them. Not sure. But they are right below the dam in pretty reliable numbers. As far as above the Comingo Dam, you'll see three little spots here. Interestingly, and no real reason that we can think of, every year, different tributary had a hit for blue catfish. This is Muddy Creek, comes in close to the top of Comingo Pond. There's a blue catfish hit there in 2019. 2020, there's a hit in Broad Creek. Don't know why. 2021, this is Conowingo Creek, which comes in on the east shore, kind of the bottom of the pond there. Not sure why, but it was a pretty strong signal. So they are above Conowingo Dam and some tributaries, but we did not find them anywhere in the pond itself. We targeted some depth samples looking for more catfish habitat. They were all clean, so we're a little bit not at a loss, but it's still kind of a mystery as to why they are showing up in these three tribs and nowhere in the pond itself.
Hey, Luanne?
Yeah.
Has it been explored? Does the DNA pass through the gut of other animals? Could birds be feeding on them?
Yeah, I have that in one of my next slides. Yeah, it's possible. It surely is. Just for completion, there's a negative blue catfish, so everywhere else was negative for blue cats, none in the tribs. Also a little surprising about blue cats in those tribs that we did see hits on, they're more of a big river fish, so you'd think they'd be in the river and not so much in the tribs. I think we're just at the mouth of each of those tribs, which is, kind of, backed up anyway, so it's not too surprising. But they weren't in any of the other, in Deer Creek or Octorara or anywhere else that we sampled. So that is the results in a nutshell. I hope that was understandable. Let me see if I can figure out how to get back to my presentation. Nope. What do you think, Mike? I do. But, nope. Yeah. That's going to be a problem. Thank you. Key takeaways. I think I went through this a little bit already. Key takeaways, though, snakeheads and blue cats are prevalent below Conowingo Dam. Snakeheads are in both Deer and Octorara creeks, kind of below their respective dams. Last year, we did find the first detection of snakeheads above Conowingo Dam. And blue cats are in some tributaries, kind of almost randomly, it seems. And we're trying to adapt our sample design every year to kind of be ahead of the invasion edge. And this has a lot of implications for fish passage. Like I mentioned, they're sorting at the fish lift. They're sorting every fish that comes up to get rid of the invasives. And it's a very kind of complex interagency collaboration. And we're leading the way in a lot of ways with this data. And every year we do it, they say, "Can you do it again next year? Can you do it again next year?" So there's a lot of interest in this, and it's been used by a lot of agencies, federal and state agencies. So it's a really collaborative effort, and I'm happy to be a part of it. Oh, there we go. Common questions. We'll go with this. So can we relate eDNA results to how many fish exactly are present? The answer is no. I can't tell you that there's, 35 blue catfish in this stream. We can do relative abundance. Like I said, they do 8 replicates. So if all 8 replicates are positive, it's a pretty good chance that they're there. Also, I talked about the cycle threshold. If it's, if it's closer to 38 or 40, it's a pretty weak signal. If it's closer to 28-ish, 30, 32, it's a strong signal. And and also the copies per liter here. I have some examples kind of our strongest DNA hits. So Snakehead, mouth of Deer Creek, 8 of 8 replicates were positive. They amplified in 33 cycles. There's 4,000 copies per liter of the DNA. So pretty strong strong signal, and, pretty solid slam dunk that they're there. Blue catfish this past year, Comingo Creek mouth, again, 8 of 8 positive, super fast cycle threshold, 29 cycles, and they amplified enough to see, and 12,000 plus copies of the DNA per liter. So, that's a strong signal. That's not coming from a bird pooping. That's what I mean by that. But yes, is it possible that DNA is there, but no living fish was present of that species? It is possible. If a bird eats a small snakehead, flies up a river and poops in a stream, and I just happen to get part of that in my sample, it's possible. It'll probably be a weaker signal, but it is possible. Or if someone caught a blue cat in the bay, took it to their house and gutted it and tossed it in the creek, I could get dead skin cells off that catfish in my sample. So it is possible. I'll give an example. This Muddy Creek blue catfish in 2019, it's only 1 of 8 replicates was positive. Real light sample. Took 38 cycles to amplify. So it's possible that someone threw a carcass out and a little bit was remaining, and I happened to get it in my sample. So we can't tell exactly, but there's kind of burden of proof. If it's 8 of 8, tons of DNA copies, quick cycling, it's probably the real fish. If it's 1 of 8 and took a long time and you never saw it again, it could be something like you mentioned, Jamie. So it's not foolproof, but it's a pretty good weight of evidence. I have one more question. There we go. And I'm often asked, how long does a DNA last in a system? It depends on flow and light conditions, because DNA can be degraded by light. So a couple of days is kind of the general guess that I've read in most papers. Not weeks, hours to days is kind of the best guess. Those are the questions I get a lot. I thought I'd preempt those questions a little bit. If you have others in mind, I'll be happy to answer them. Here is me standing in the first flowing section of the Susquehanna, 1 mile south of Otsego Lake. That's how small it is. I was very happy to be there. So any questions? I'll be happy to answer any questions, or we can wrap it up.
Hey, Luanne.
Yeah.
Sorry, my dog starts barking as soon as I come off mute. But
I thought you weren't coming to the presentation, Ben.
No, I changed my plans for you.
Oh, thank you.
So I have like 5 questions teed up here.
I'm ready.
The
Hit me.
first answer, the persistence or how long eDNA lasts in the system. That was a good one. I was wondering about the recommendation for monitoring in the upper set at 3 years. Is that time frame sort of consistent with what would expect a population to establish itself, or there would be, seems to me, for me, it seems like maybe we should be doing it every year, but I don't know. So I'm curious about that.
Kind of arbitrary on my part. Just kind of, allocating resources to where they're a little more prevalent. Obviously, if we hear of a record or a catch of one of those species up there, we could certainly do another round sooner than that. But 3 years was just my best guess as to when it might be important to try to look at it again.
Mm-hmm. And then what about once we do find it, what sort of mitigation measures are available, or are we looking to do any sort of, I don't know, once we have invasive species in the system, what do you do about it? What can you do about it, if anything?
It's Aaron's problem after that. I just did this part. That's Aaron's problem. No, I'm kidding. It's hard to stop an invasion, certainly. There was a case in Delaware, Delaware Basin in New York State, where there was some snakeheads. They did a massive eradication, like rote-noting streams and killing all the fish and getting rid of them. And they did eradicate that small population of snakeheads from that stream. That's pretty extreme. There aren't a lot of good ways to do it. So the management is kind of mostly just keeping tabs, to be honest. Like you really can't, unless it's very small and isolated, it's hard to get rid of them once they get started.
Well, thanks. I have one more quick one. I'm wondering about the middle Susquehanna. Are you guys going to be looking there too, or?
In the middle of Susquehanna,
Yeah.
I don't know. Is there a problem there I should know about?
No, I mean, you hit the upper and the lower. I just wondering if there's a reason you didn't go to the middle. Or
I'd be happy to if there's money. I don't know. I've not been made aware of a need at the moment, but I mean, there's certainly probably reasons to think that they might be exposed in that area too. I don't know.
Cool stuff. Thanks for presenting.
Sure. Thanks for making it work for your schedule.
Luanne,
Yeah.
just to build on Ben's question, I assume, like on an earlier slide, we showed the Mohawk proximity to the upper. That's probably, I assume that's why you targeted that, because there's a prevalence that, much like in the lower Susquehanna, snakeheads are below the dam, so you're just sort of trying to.
Yes. If they're close by, we're going to try to keep tabs and see if they're getting into our waters.
Yeah.
You know? Yeah. And those two species were on the New York DEC's like kind of tier 1 high risk invasive species. That's why we chose those two in particular. We could choose any, and so blue catfish for that matter. But those two were on kind of their highest priority risk level for that part of New York State. Okay.
Oh, go ahead, Jamie.
Go ahead, Andy.
Okay, no, quick question. I was just wondering, with this new technology, I was wondering, we've only been in it a couple years, but what's sort of been the trend, like with the cost and accessibility, turnaround times, reliability? Like, is it moving slow or is it moving fast, or?
It's hard to say because of COVID kind of slowed a lot of things down.
Oh, yeah.
So the first slowdown in 2019. There was already a marker created for snakeheads that was already available to the lab to use. But for blue cats, they had to actually create a marker to do the test with. So they had to do all the validation of testing, make sure it wouldn't amplify channel catfish, or wouldn't amplify flathead catfish. It would just target the blue catfish genome. So it took a lot of work to get that marker validated when they run our samples. So that was the kind of a slowdown in 2019, and then COVID. But this past year, we gave them the samples, I think, in July and got the results back in late October. So it's a couple of months turnaround time. I think costs have been fairly steady. The lab costs are about $10,000 for like these 50-ish samples, around that. And they give us the supplies to do it with and everything. We just have to give them the filters back with DNA on them. So they've been pretty standard. I know that lab in Lamar, they're taking samples from everywhere. So they had kind of a big backlog, like 2018, 2019. Now they're kind of getting through them a little faster, but there's definitely a backlog in the beginning. But they do everything I've ever heard of almost. They do that. Lab goes through their lab. The log perch stuff we're doing, the Goby stuff in the Mohawk, our work. I heard something else recently too, but yeah, that's used very, very widely. Their lab is.
Yeah, I was going to ask, you mentioned in the library the log perch. So besides the invasive, things like rare or threatened, or even the eels, like you mentioned at the outset, labor-wise, whatever the cost of this is, it's nothing compared to taking crews out and doing 2 sites a day.
Yeah.
So, things like hellbender, log perch, or doing eel census, are you or Aaron or your colleagues, using these techniques for those kind of outcomes?
We have thought about maybe for eel. We have thought about it a little bit, and I've discussed it, not recently, but we have. There's an eel marker that's well developed that it could be used pretty easily. I think there's a log perch one developed for the work they're doing with the reintroduction. So we haven't used it for any other applications besides these 3 invasives, but we certainly could. It's possible. It's the same technique. It would just be different marker. We haven't, not yet. Jason?
So, eDNA could be in the air, water, soil. So, you say in the water column it doesn't stay. Does it grade in the water column, or does it go into the sediments
And have you tried sampling sediments for eDNA, or is that not fruitful?
Good question. It often is in the water column, so water's going fast, it just kind of moves downstream and goes into the bay or the ocean or whatever. If the water's going slow, it can drop out and get to the sediments. It can also be degraded by light, so if it's in a slow moving water body, it can be, too much light can make it unusable. But yes, people do look at eDNA in sediments. I never have. I don't know what the process is. I assume it's similar-ish. You have to get the DNA out somehow. I don't know how you filter it or get it there, but people have done it, and it is viable, but I've never done it. We never tried it.
Another methods question, like, for 2 liters you say you sample, obviously, and that's a good amount of water on it. Is there any, like, way to increase the volume, or is there passive samplers for eDNA, or is that foresight just not work with that?
There, 2 liters is kind of the standard that they use for fish and wildlife service because it can be filtered in a reasonable amount of time.
Right.
It can be filtered in about a half hour to filter a 2 liter sample. So we have a manifold, we have 4 going at one time, so we can do 4 in, I don't know, half hour, 45 minutes, basically. But there are passive, also, if you do turbine water, or like in Virginia, that black water areas in the swamps, I've heard people, like, strap their filter into their car seats and their seat belts and drive because it's taking 8 hours to get through the water through a filter. So we're lucky we have pretty clean water, not that turbid. But they use you can do more, and you might get more DNA. You know, it's possible. You don't see, but I think 2 liters is kind of the standard practical volume that can be used in a practical sense. There are passive samplers. I got a pitch for one at a conference a couple of years ago. There's pros and cons. Basically, it's like a big fishing bobber, and it has a filter in it, and you just put it in the water, and water passes through it for, a week or a month or whatever, and you go back, and you just sit in the lab the entire thing. There's no transfer, there's no chance to get it messed up or, contaminated or whatever. But it can get clogged. If you have it over a month, you don't know if something was just passing through, or if it's recent, or you don't have a time frame on it. But there are certainly applications of passive samplers that are out there, yeah.
Is there a record of what constitutes native versus non-native as far as how far back our records go? And I realize that that's a very generic question, and probably there's not a simple answer, and then my follow-up question would be, is at a certain point in time, would blue catfish be considered, at what point did they become considered native? Because I guess what Ben was saying is, or what you're saying is, it's obviously easier to try to stop them before they get here, because once they're here, it's like, forget it.
Yeah, Aaron might have a better answer, but I'll try to answer it. Like, for instance, brown trout are not native to Susquehanna, but they're, like, naturalized, and they've been here for so long, and we still mark them as non-native because they're not native.
Smallmouth bass, too.
Same thing, smallmouth bass are not either. So, brook trout are our only native trout to the Susquehanna. Rainbow trout, brown trout, not native. But they're here, and they reproduce wildly naturally, so they're kind of naturalized in the system. So, and we do have a list somewhere, Aaron can probably give it to you, of the species that are native to our drainage, that we know, by all countable records, that we know are native to our drainage. But there's some, like, brown trout that are naturalized that we think are okay. And I don't know, in 50 years, will snakeheads be naturalized, and we'll be like, not a big deal, they're just here. I don't know, I'm not sure when that cutoff is, or when you decide. Other, for instance, there's 2 non-native darter species, which we find all over the basin, but they're not invasive, but they are non-native. They're from the Ohio. We have like 3 native species of darters, or 4, and then 2 green-sided banded, which are not from here, but we find them all the time, and they're not hurting anything, but we find them all the time. So I'm not sure when that line is when they become naturalized, or just think, well, they're here, whatever. I'm not sure where that line is. Aaron's here, wants to chime in and correct me, feel free. He's a fish expert.
Well, the naturalization is just, that's like a societal concept and abstract, just of whenever, people aren't bothered by it, more or less. But as far as determining the actual native status of fish, there's a lot of you can do that through genetics. There's a whole field of zoogeography that can study, through historical records and otherwise, where fish have come from, through time, through, the evolution of Earth. There's a pretty good fossil record, and even through the glaciation recently, we know where fish came from and likely how certain ones show up in different drainages and why, due to stream captures and things like that. So there's a lot of good evidence for it. Great talk, by the way.
Thanks. Anybody else? Good question.
I have one more.
Sure.
And maybe we don't know this because it's just evolving now, but how sophisticated is the eDNA as to if one of the non-native species was to mate with a native species? Are you able to tell that happened? Are you able, like, if you were to extract the eDNA from
That's a good question.
product of that?
I don't know a great answer for that. I do know that DNA target is in the mitochondria and it's passed maternally. So if the blue cat was the mother, it would show up as the hybrid would have the blue cat I was looking for. It would catch it.
Oh, wow.
But I don't know anything else more than that. Like, so it's passed maternally. Our targeted genome is a mitochondrial CO1 something gene, and it's maternally passed. So the mother's a blue cat. It would show the blue cat.
I'm assuming that for especially the snakehead, if it's that invasive that quickly that when they reach full size, there aren't natural predators. Is that one of the? I mean, is there anything that eats this stuff like when they're babies? You guys, maybe this is an Aaron question. Like, just not. I'm assuming the volume's just not enough. There's nothing that kills them.
No, nothing from when they're big as far as another fish. I don't think. I mean, maybe like a muskie or a walleye might eat them.
A blue cat.
Blue cat might eat them. Yeah.
Another invasive.
But birds, there are birds do birds do eat them.
Okay.
We have evidence of herons with small snakeheads in their mouth. So there is some bird predation certainly, probably some small mammal predation on small snakeheads. Once they're big, they don't have a lot of natural fish predators.
Well, I was just thinking that maybe it's one of the, because I know like in farming there are a couple invasive species of plants I still refer to as invasive because they still cause a lot of problems. So maybe does it keep being called invasive mostly because they stay a problem?
Yeah.
Like brown trout, the way that, just from my day with you guys, don't seem to be a massive problem, whereas these things are, right, they're non-native when they're not a problem, they're invasive when.
Correct. It's a behavioral term, not like a.
So if it balanced out, like if something developed that would start eating these things and knock them down, then that might be a time when, like it might evolve to just being a non-native, like, oh yeah.
Yeah, correct.
Is it something, that's, okay.
Yeah, it'll take time, but yeah, it's possible that would certainly kind of how it might evolve. Anybody else? Go ahead.
No, did someone else have a question?
Nope, I was just wrapping up. Go, Ben.
Well, I just want to know how Curtis gets a day with you guys, and I've been asking for like years to come out with you guys, and I don't get an invite.
Wasn't my work.
It was a performance objective. I was required to do it.
Okay, John.
I've asked to go again, and
You want that? Love to have it.
We got to write that in.
I want a hellbender day, and I still haven't gotten that. I would like one of you to find me a hellbender, so.
They're pretty rare. So I'll just wrap it up. Thanks again to Drew and the commission, especially for putting all this work and Drew being supportive of it. And it's been pretty cool, and it's kind of fun to kind of cutting edge of technology and monitoring applications. And I think it's a great fit for us to do this kind of work. So thanks for everyone for coming out.
Great stuff.